Again, welcome everyone to today's webinar. My name is Layla and I'm the Community Engagement Coordinator for the Canadian Cancer Survivor Network. And today's webinar will be about tumor agnostic treatments and the future of specialized medicine. So we are pleased to welcome our three incredible presenters for today's webinar. We are joined by Dr. Poul Sorensen, who is a board-certified anatomic pathologist specializing in the molecular pathology of pediatric cancers. He undertook his undergraduate, medical, and PhD degrees at the University of British Columbia and McGill University. We also have Dr. Zulfiqar, who, after postgraduate training at the University of Kansas Medical Center, joined Eastern Health Cancer Center Cancer Care Program in St. John in 2003. He is currently a medical oncologist at the BC Cancer Agency in Abbotsford and is a clinical associate professor of medicine at UBC. And lastly, we have Ted Taylor. He was the vice president of Infinity Power Products prior to his diagnosis. He has recently been involved in advocacy efforts and is working on a new website called Glioblastomaguide.com and a YouTube channel for people diagnosed with glioblastoma, where he provides resources, information about treatments, and where to find experts. In this webinar, we will first hear from Dr. Sorensen about his discovery and how it contributed to the development of tumor-agnostic treatments. Dr. Zulfiqar, a medical oncologist, will then further discuss TRK fusion cancers and how he has been able to treat patients. And then we'll hear from Ted about his diagnosis and his experiences being treated with Vitrakvi. At the end of the presentation, our speakers will be able to answer your questions. However, at any time during the webinar, please feel free to type your questions into the questions tab on your control panel. For those of you who have not attended an earlier webinar, the Canadian Cancer Survivor Network is an organization working with cancer patients and survivors to learn about health system complexities, connect with others to plan action, and act to promote best care and healthier survivorship. If you would like to learn more about CCSN, please visit our website at survivornet.ca. This webinar is being recorded and it will be available tomorrow on YouTube. In addition, slides will be available on SlideShare and links to both will be sent to the email you provided at registration. I will now turn things over to our presenters. So welcome, Dr. Sorensen.
Thank you very much. Just going to make sure that you can see my screen and just let me know that you can. Layla, are we good to go?
We are good to go.
All right. So thank you very much for this kind of invitation. I'm a research pathologist at the University of British Columbia, and my research lab is at the BC Cancer Agency, and this is the view from the top of the building. Unfortunately, my students like to go up to the top of the building and look at the view, sometimes more than doing the work, but at least it's a beautiful place to, to work from. So I'm going to tell you a little bit about how we came to discover a class of tumor drivers known as NTRK fusions. And I apologize in advance if the presentation gets a little bit technical, but maybe we can clarify things in the question period. So just as an introduction, I think many of you know that for decades, patients have been treated with chemotherapy and radiation, and many of these anti-cancer drugs target dividing cells, sort of in an empirical fashion. And so they may be highly effective in some patients, but less effective in others. It's sort of shooting lots of arrows at the target, but this also causes a lot of risks and side effects. So as we started to learn more about the molecular drivers, the mutations and other genetic alterations that drive malignant disease, we started to identify so-called biomarkers or altered genes that can actually be targeted and therefore predict a patient's response to therapy. And in this way, patients are more likely to get therapies with the greatest impact and the least side effects. And today's presentation is about that. So to put this another way, this has all led to something called personalized oncology or precision oncology, where you incorporate genetic information into treatment decision making. So in the old days, pathologists would look through the microscope. I'm using the example of lung cancer here. Identify malignant cells, declare the biopsy as suggesting lung cancer, and then the patient was put on chemotherapy. And this would work for some of the patients, but not all the patients, and then there would be side effects. Keeping with the example of lung cancer, once we started to identify some of the altered genes that were actually, the proteins of which were driving the disease, then researchers and pharmaceutical companies could actually develop drugs that could target each of these individual alterations. And in that way, we could separate patients out into different groups that were more likely to respond to these specific therapies with less side effects. And we've even gotten more sophisticated now, where we can potentially take a blood sample, look for a mutation in a patient that's already received therapy, and identify some kind of mutation, additional mutation that may confer resistance to a particular drug. And then we can come in with another drug based on that information. So this has been really great for what we call precision oncology. So we live on the West Coast, so I have to use a skiing analogy. What we've learned by this kind of analysis is that there are a lot of different pathways that tumor cells can use to get down the mountain. And so this kind of makes it difficult to come in with only one drug, because sometimes they can switch to a different run. So wouldn't it be something if a particular tumor had only one way to get down the mountain? This is, of course, a luge, could be a bobsled track. But if we could block that one pathway, then maybe we have a much more efficient way to target tumor cells. And just to remind you, and I apologize because this is a little complicated, but when we look for the major types of genomic or genetic alterations in cancer, they're kind of divided into the group on the left. These are mutations where a single base pair is altered, and this can either turn the expression of a gene off or create an activated gene that can drive the malignant process. Sometimes little bits of sequence are lost from a gene or gained from a gene. And those typically turn off a gene, but they can also turn on a gene or create a protein with hyperactivity. Sometimes there is what's called copy number amplification, where a region of a chromosome becomes duplicated or triplicated, and then you get more doses of the gene that's located in that region. What we were interested in many years is this concept of a rearrangement, where pieces of different chromosomes switch places. This is called a chromosomal translocation. And this actually generates what's called a gene fusion, whereby parts of one gene become fused to parts of another gene to create what's called a chimeric gene. And these chimeric genes can then have new functions in the cell, and these functions are often or can be pro-oncogenic. In other words, by creating a new gene, these genes can be oncogenic in different contexts. And this brings me to the discovery of the first recurrent example of a genetic alteration involving these NTRK kinases that we're going to talk about. This is the discovery of the ETV6 NTRK3 fusion that we made many years ago in pediatric soft tissue sarcomas. So this all started out very innocently. I'm a pathologist, and sometimes you're shown a case that you really can't figure out what it is because it looks very much like other things. So this is a perfect example of that. By microscopy, a disease called benign fibromatosis that can happen in very, very young kids, under 1 or 2 years of age, can occur. These are benign lesions that can be treated with surgery alone. However, there is a malignant version called congenital fibrosarcoma, and sometimes you don't know whether a case is the benign version or the malignant version. And so we asked the question, could we find some sort of chromosomal marker for the malignant disease, or some sort of molecular marker that clinicians could use to differentiate these two entities? Because you don't want to put a 1-year-old child on chemotherapy or some other or radiation if you don't have to. So we needed something to differentiate these two entities. So I wouldn't be telling you this unless we did. And so over 20 years ago, we noticed that in the malignant version, there was a small, very subtle change of chromosomes. This is chromosome 12 and 15, and there was an exchange of material. This was extremely subtle, but I had a very good summer student in the lab that decided to go after this. And to make a long story short, it turns out that this chromosomal rearrangement resulted in a part of a gene from chromosome 12 called ETV6 becoming fused to a kinase portion of a gene called NTRK3. And what these kinases do, they're enzymes that by their activity, they can turn on pathways inside cells. And so without going into details, the fusion of these two sequences together created a kinase that didn't need to get turned on by some sort of external growth factor. It could automatically mediate cell survival and cell growth of cells expressing it. In other words, it could induce oncogenic transformation. So when you see this sort of thing, you have to do some basic studies. And so what we did is we took this, you can recombinantly express any kind of gene in other cells. And so we used mouse fibroblasts, and we noticed that when we put this fusion gene into these cells, they changed their appearance. They started to grow faster, and they became much more what we call refractile. So that was a first hint that this was actually oncogenic. And then we put it into this gel-like solution, whereas normal cells will just sit there or will die. These cells all by themselves started to form these clusters. It's another word for this is anchorage independent growth. And then finally, when we put them in mice, the cells in mice, they formed tumors. So these were all big hints that this was an oncogenic driver. So then we made a very surprising discovery that the same alteration, this ETV6 NTRK3 fusion, was also present in a subtype of breast cancers. And this is something called human secretory breast cancer, which occurs in a much younger age group than more typical intraductal carcinoma. So this was interesting, a bit puzzling. And then other people started to find the same gene fusion in completely different tumors. This is an example of something called a mammary analog secretory carcinoma of salivary glands. So here we had a situation where this ETV6 gene, which we thought was a driver of pediatric sarcomas, is now also expressed in secretory breast cancer and a salivary gland tumor, even though these have look differently under the microscope and are located at different anatomic locations. So we had to prove using a whole new strategy that this was actually an oncogenic driver. So we teamed up with a group at Harvard led by Stu Orkin, and we made a mouse in which this gene was programmed into the cells of the mice, in the breast cells of the mice. And these mice, about 100% of these mice developed breast cancer. And what was really interesting about these breast cancers, and I know you're not pathologists, but just take my word for it, these mice developed a lot of the precursor lesions for breast cancer, something called hyperplasia, carcinoma in situ, and early invasive breast cancer. So this was really evidence that all by itself, this ETV6 NTRK3 protein could drive malignant transformation. So it turns out that there are two other family members of this types of kinase. So this is something called a kinase tree, where the evolutionary biologists have linked different proteins together. And these NTRKs 1, 2, and 3 are also known as TRK A, B, and C. But what they do in normal central nervous system tissue is they react to growth factors, and then because they're enzymes, they can turn on different processes in the CNS tissue like pain, thermal regulation, movement, memory, appetite, metabolism, and proprioception. So what happens in these fusions is that the working and the enzyme end of it is still there, but now they don't need these growth factors to be turned on. And so that's how they work. They're so-called ligand-independent enzymes. So they're not controlled by the regular control mechanisms that normal cells have. So then something called next generation sequencing started to happen, where many groups started to do sequencing, not the old-fashioned way of looking at genetic alterations that we did. And people started to find other examples involving all three of these NTRK genes. The most common one was still the one we found, But then other ones started to come in. And as you can see here on the right, these were present in many different tumor types. So the incidence of these types of fusions were way more, was way higher than we had previously anticipated. For example, here's an example of something called an LMNA NTRK1 fusion in a soft tissue sarcoma in a 41-year-old female. And so it turns out that these NTRK fusions involve many different partner genes and in many different diseases with high clinical unmet need. And sometimes, like the example I gave you in congenital fibrosarcoma where we first discovered it, they're present in about 100% of cases, similar to this mammary analog, secretory cancer or secretory breast cancer. Sometimes they're present in a smaller proportion. It's a very interesting story that after the Chernobyl accident, about 14 to 15% of especially young patients develop papillary thyroid cancers, and they had this ETV6 NTRK3 fusion. And then some other diseases such as glioblastoma multiforme, the incidence is 1%, and in others it's sort of 10%. So it's a very interesting composite of all these incidences. So finally, just to summarize all of this, it turns out that all three NTRK genes are implicated in oncogenic fusions, and now there are at least 25 different tumor types that have had these NTRK fusions identified, including breast cancer, ovarian cancer, colorectal cancer, melanoma, and lung cancer, and some of the other ones that I mentioned. They're now implicated in up to 1% of all solid tumor malignancies. And these fusions all result from these translocation events, chromosomal translocations that I mentioned. They all result in overexpression of the enzyme and this ligand-independent activation, which I mentioned to you before. They can turn on these pathways without needing some kind of hormonal stimulus. And so, of course, this suggested to the pharmaceutical industry the possibility of developing a drug that could target all three of these NTRKs with the same drug. And that, of course, has happened with Larotrectinib, or Vitrakvi, as it's now called. And I thought this was a good place to transition over to the next speaker to tell you, as a medical oncologist, some of his experiences with this agent. So I thank you for your attention. Again, we can come back to this, to some questions a little bit later. Okay.
All right, thank you very much. Thank you, Dr. Sorensen, for not only this comprehensive review, but also for all that work you have done, paving way for clinical trials and eventually saving lives. I'm just going to talk about the clinician aspect of tumor agnostic treatments. Traditionally, how we have seen cancer is not as a single disease, but a heterogeneous combination of diseases. Even when we see cancer arising from one single tissue, it behaves differently depending on the epigenetics, depending on the tumor mutational burdens and driver mutations, and the interaction with the host. And therefore, how we have come to design treatments is really based on phase 3 clinical trials. And in phase 3 clinical trials, the attempt is to have maximally matched some of the characteristics of the patients who are going through it with a similar comorbidity, similar performance data, similar organ functions, and have the same sort of tumors as much as possible with the staging as well as some biomarker stratification to be studied in parallel to each other, so that the comparison would be less biased and we may be able to generate a knowledge about which treatment is more effective or less effective compared to the current standards of treatment. That's how the traditional chemotherapeutic medications have come into existence, informing us that certain combination of cytotoxic chemotherapy work in certain types of cancers and it doesn't work in other types of cancers. some of those examples I've highlighted in these slides. Targeting the DNA synthesis or interrupting the tumor DNA by one example is 5-fluorouracil, which is affecting the rate-limiting enzyme of thymidylate synthetase. Oxaliplatin is an example where it forms heavy DNA adducts in the cancer DNA, and irinotecan as a topoisomerase inhibitor, blocking the uncoiling of these DNA, eventually disrupting cancer DNA with the hope for decreasing the effectiveness of growth and reducing the tumor burden and improving the person's survival, and possibly in certain situations putting a person into complete remission and potential cure. Other fronts, other aspects from where the traditional chemotherapies have been used is attacking the mitotic spindle so that cancer cell doesn't divide, using anti-metabolites like nitrous ureas, or starving the cancer by inhibiting the angiogenesis. So all the cancers need new blood vessels, and those neoangiogenesis can be targeted, and by that we can reduce the tumor growth. And in complement with other cytotoxic treatments, these anti-angiogenic treatments have been shown to improve survival. However, as you can see, that these are all tumor site specific, and they're tissue site specific treatments. And what we are talking today is about tumor agnostic treatment. How does tumor agnostic treatments differ? This is a treatment when a drug is used to treat any kind of cancer, regardless of its origin. So if we have a proper type of molecular alteration and a drug that can be effective against a specific molecular alteration, then that drug can work regardless of where in the body the cancer had started. And as Dr. Sorensen eloquently pointed out, that one of these alterations is NTRK gene fusion, which is found in mostly more common in pediatric population compared to adult population, and the incidence in glioma patients is between 0.6 to 1.4%. And similarly, as you can see, all the other solid tumors doesn't have a very high incidence of NTRK fusion positivity, except the secretory breast cancer. On the other hand, pediatric population have higher incidence of NTRK fusion, ranging 92, 83, 91%, depending on the type of the tumor. So I wouldn't go into detail. Dr. Sorensen had a much refined analysis of how the gene fusion takes place. This is a very simplistic view where part of the chromosome A and chromosome B are fused through either translocation, inversion, insertion, or deletion. They're different mechanism. And when this sort of fusion happen at NTRK 1, 2, or 3, it end up resulting in activation of pro-differentiation genes and pro-survival genes, leading to tumorigenesis without any need for any growth factor. And Larotrectinib is one of those medications which is able to bind with the NTRK kinase domain, and therefore by inhibiting that signaling would prevent the downstream anti-tumor effect, leading to responses, improvement in progression-free survival, overall survival. However, as we talked briefly about how phase 3 clinical trials work, is by having a certain number of patients available to have a proper statistical power to have a valid analysis, so that we can make a proper decisions which are generalizable for majority of people in the face of uncertainty. However, as we know that in NTRK fusion, the incidence is so low that it would be almost impossible to conduct similar type of phase 3 prospective randomized controlled trials. And therefore, this existence of basket design is there. And to make it very simple, if we have 2 patients out of 100 melanoma patients who have this fusion positivity, 1 in 100 of thyroid and 2 in colorectal cancer, then we pick all those molecular alterations and put them in one basket. So in that basket, we have disease agnostic basket. This is not a tumor specific basket. People in here have different types of cancer, but they have same molecular alteration, which is NTRK gene fusion. And that's how prospectively that we can study that drug that will target that molecular alteration. One such study with expanded analysis of this data was presented in European Society of Medical Oncology Virtual Congress in 2020. And in this expanded analysis, a contribution came from all the top institutes in Europe and US, and presentation was done by St. Vincent University Hospital and Cancer Trials in Ireland. And this analysis showed us that the Larotrectinib had demonstrated about 79% overall response rate and 28.3 months of impressive progression-free survival with 44.4 months of median overall survival in people who had TRK fusion gene positive cancers. And now currently, this is approved in 40 countries in European Union, including US and Canada. Just quickly going through that pooled expanded analysis of phase 1 adult and phase 1 and 2 pediatric and adult and adolescent population together, there was 175 patients with non-primary CNS TRK fusion cancers. And they had TRK fusion determined by accredited labs. And adult dose of Larotrectinib was 100 milligram BID and pediatric dose was 100 milligram per meter square. The primary endpoint of analysis was overall response rate, and secondary endpoints was duration of response, progression-free survival, overall survival, and safety. So as we see, the majority of these patients had sarcoma, followed by thyroid, salivary, and lung and colon cancer. And there were other combination of different tumors in there. I want to bring in your attention to the most important aspect here is that majority, almost half of these patients, 46% of them, had prior 3 lines of chemotherapy. So as a clinician, we know that in advanced cancer who have resistance to first, second, and third line of treatment, the disease is advanced already enough, and the cancer has acquired those molecular adaptations where they become quite resistant to subsequent treatments. So having at least half of those patients who had more than 3 lines of treatment makes it a tough population to treat. And in this analysis, majority of people were positive for NTRK1 and 3 gene fusion, and NTRK2 was in 6 patients. So having said that 46% of people had more than 3 lines of treatment, it is so impressive to see this response that at objective response rate was 78%, which was previously unheard of in this population. And in adult population, 71%, a little bit higher in the pediatric population. And not only that, people were able to continue the treatment for longer periods of time, possibly because they are able to tolerate it better, disease burden was decreasing, their symptoms are improving, and the drug did not have much of the side effects. So currently, the range of the duration of treatment was from 0.1 to 51.6 plus months, because they are still ongoing treatment with Larotrectinib when the data was reported. The duration of response at 12 months, 81% were still responding. At 24 months, 61%, 66% were still responding. And if, as far as progression-free survival was concerned, at 12 months, 69% of people who responded, they did not have any further progression. And at 24 months, 61% did not have any further growth of the cancer for which they were being treated. Overall response rate was also quite impressive, with 12 months, 90% overall survival, and 24 months, 83% overall survival. Having said that, if I am a cancer patient, I not only want to live longer, but I would also want to feel better so that I can enjoy the quality of life. Therefore, this analysis was very important. Before we quickly go through this, it is important to understand that adverse events are defined as any adverse event which may or may not be related to treatment. So if we are seeing fatigue, cough, constipation, diarrhea, dizziness, myalgias, all of these side effects or adverse events may not necessarily be due to treatment. It may be also related to the disease itself. So all of these adverse events are reported, but important thing to look in there is that the treatment-related adverse events mainly were grade 1 and grade 2. Grade 3, where people needed some interruption and needed some hospitalization, they were very few. Grade 3 treatment-related adverse events were 14%, and grade 4 treatment-related adverse events were 2%. And only 11% required dose reduction because of adverse events, and dose reduction because of treatment-related adverse events were only 1%. And only 2% of people discontinued treatment because of treatment-related adverse events. So it is, in conclusion, this expanded analysis showed us that a response to Larotrectinib was rapid, durable, and the median progression-free survival for more than 3 years is quite impressive in this population of patients. And Larotrectinib continues to demonstrate a favorable long-term safety profile, and the data is sufficient enough and convincing enough for it to be used as therapeutic target, and routine screening and testing of patients with NTRK gene fusion is warranted. However, as you can see that in order to detect one, you have to test 100 in glioma patients. So there is an ongoing work to identify clinical and biochemical characteristics where there may be better targeting those patients who may likely have more richer and more prevalent NTRK gene fusion positivity. So I will stop here for inviting Ted to tell us about his experience.
Excellent. Okay, just give me one moment here. Okay. Well, I'd like to first thank the Canadian Cancer Survivor Network and all those involved in making the webinar today possible. My name is Ted Taylor, and I'm a glioblastoma multiforme survivor, and this is my presentation on Miracle in the Making. The diagnosis. For me, I had to go into the emergency on 2 separate occasions because I've been having severe headaches, nausea, and vomiting. This went on for a period of several weeks. They sent me home on each occasion. I went in to go see my doctor, and he said the same thing and sent me home, just trying to get rid of the severe headaches and whatnot. These were not like normal headaches that I was having, but try and take some Tylenol or Advil, something like that. It got so bad after about 3 to 3.5 weeks that I had lost about 20 pounds at this point, and I was no longer able to keep food or drink down. We went into the emergency room and this time insisted on a scan. At that point, we received the bad news, and you can see there's a very large tumor about the size of a large egg. And it was even pushing, you can see the midline here, right down the center, was being pushed over from the right to the left side. And at this point, it was so serious that they suggested that I go see the surgeon the next day. We went in and saw the surgeon, and he said that the situation was very dire indeed, and that I was to have surgery 6 days later on October 31st of 2018, because apparently that's the best day on Halloween to have brain surgery. Once I was out and finished with the surgery the following day, I began my research in earnest. This was a very difficult time because basically all the information said that this was it. You're likely going to just die here. And as a single father with 3 children, that was not something that was sitting well with me. I did research every day, and on November 26th, I decided to take a break at one point, which was very rare, and I sat down, turned the TV on, and the last channel it was on happened to be the news. So that popped up, and the next segment that came up immediately was that the FDA had just approved the drug Larotrectinib. This was incredible. As I saw on there, a list started showing up of all the different types of cancers that it could potentially treat. And one of them was glioblastoma. I hit the record button, watched it a couple of times, and then I decided at that point that I needed to dive into doing some research. I quickly discovered that only about 1% of patients will actually have the NTRK1, 2, or 3 fusion. At this point, I knew that I had to get onto this, and found I went to talk to my oncologists. I brought it to them, and they had not seen this before as far as a drug. It was not available in Canada, but step one was that I needed genetic testing. One of my oncologists suggested that I should talk to Foundation One as a potential. That was very expensive as an option. One of my other oncologists, he looked into it, and he was able to actually get some preliminary testing done locally. When we did that, we got some results back, but it was a bit confusing because both the foreground and background material both showed that it was NTRK, but we did not know what that meant. At that point, we submitted to Foundation One to get that process underway. My oncologist teamed up with his pharmacist and put in an application to Health Canada on my behalf to have this drug approved. Health Canada, in the approval process, Health Canada did approve me under what's called the Special Access Program as an initial patient for Larotrectinib. Bayer required the Foundation One report. And fortunately, because we already had this in the process, only a few days later, that was actually able to come through. And at that point, Bayer did approve me as I was NTRK1 positive. This was amazing. The process then moved to getting the drug, and it was shipped from the United Kingdom to Germany and then to Vancouver. And at that point, my dad and I were actually waiting with the pharmacist at her location. And the package came in, it was in a special box, and we opened it up. I received the instructions on how to use the drug, and it was the capsule form. And I began taking it of all days on Good Friday of 2019. Hope. This drug has created hope. The efficacy is 75% in the initial trials, where it either stops the cancer from growing, or it can even reduce it, or in 22% of those people, you get a complete initial response. I can tell you there's never been anything like that, especially in glioblastoma. There's no medications. As a comparison, temozolomide, which is the chemotherapy drug for glioblastoma patients, was the largest breakthrough in 15 years, and it provides a few extra weeks of median survival. Under the standard of care in my case, I received a diagnosis of 14.6 months for my median survival, and that's with all of the advancements and achievements in human history up to that point. In most cases, optimal treatment for glioblastoma patients have a median survival of less than 1 year, and there is no cure and a very high rate of recurrence. Only 25% of patients survive more than a year, and the US National Center for Biotechnology Information lists long-term survival beyond 3 years limited to less than 2%. In other words, you have a 98% chance that you will not make it. Side effects and adjustments. I take Vitrakvi 2 times a day with breakfast and dinner daily. The side effects in my case were that my liver enzymes increased, my AST and ALT, and they went up significantly. Now, I had read in my research that in the trials, some people even had to stop if they had their ALT and AST go up too high. So be your own best advocate. What did I do? I started doing some research. I looked into this and I found that there's a couple of different mechanisms that could possibly make a difference. One of them was water intake, and the other one was through bilirubins in my blood. So what I did was I adjusted. I had been taking 3 liters of water a day. I changed this to 4 liters of water a day, and the result was that on my next blood test, my ALT and AST had come down rather significantly. I decided to adjust this to 5 liters of water a day and see what that would do on my next blood test. It actually started going back up again. So I knew at this point I was probably overtasking my liver a bit. So I dropped down to 4 liters a day, and that's where I've kept it since. And the results have been very good. Also, sun exposure increases bilirubins, and that also made an impact. Now I can show you the results of that. This is a graph of what my ALT was before water adjustment and my bilirubins. And you can see over here, it was going parabolic. That meant that I was likely going to be taking off the drug. The AST in the same result. After the adjustments, I was back into the normal range or slightly above. Again, be your own best advocate. You can see here, just slightly above. Side effects and adjustments. If we juxtapose these results, which are just, some increases in my ALT and AST, against the standard of care, we can see that my reaction to temozolomide, again, the chemotherapy drug for glioblastoma, it completely destroyed my immune system. My red, white, and platelet blood cells were absolutely level, to the point where my platelets, which should normally be in a range of between 150 to 400, mine got down to 19. I needed 6 emergency platelet transfusions over the next several weeks. With radiation, I received 60 grays of radiation. And the day before I was to start my radiation treatment, my radiation oncologist called me up and spoke to me and said he thinks that we should increase the area that we already had around the tumor by an extra 2.5 centimeters of radius. And because the disease is so aggressive, that this would give us the best chance. I was told the implication of that is that I would lose the hearing in my right ear, 100%. That was very difficult to hear. But again, looking at my situation, that was the right decision, so we decided to go ahead. At that point, on December 31st of 2018, I did lose the hearing in my right ear. Now, I prayed that night, and the next day when I woke up, my hearing was restored, and I have actually had that ever since. So this is part of my miracle. Other results of the treatments that I've received: fatigue, drop in stamina. My memory is not as sharp as it used to. Sometimes I'm hunting for words. I'm not the same person, but I'm blessed to be alive. I'm able to keep calm and have my friendly personality, and this was not a guarantee after the radiation treatment. Liquid versus the capsule. On April 19, 2019, as we discussed, I started taking the capsule form of Vitrakvi. This was great. On September 7th of 2019, I started the liquid. This is because Health Canada approved the drug for Canada on July 31st, 2019, but the approval was only for the liquid version. Now, I switched over to the liquid, and the purpose of this was because it's much easier to adjust the dose in pediatric patients and others where they need an adjusted dose. However, the practical impact for adults is very different. It anchored me to my home, and as a single father with 3 kids, that meant that we couldn't go on outings or do certain things like that because the liquid form has to be kept refrigerated in a very fine corridor. So it can't get too cold and it can't get too warm. I also kept the drug in 2 separate locations. So I had 1 bottle at my home and another at a separate location because we get fall and winter storms and the power could go out for extended periods of time, and I didn't want to lose all of the medication. So there are different implications between them. I worked with Health Canada and with some advocacy groups towards having the capsule being approved. And fortunately, I'm back to the capsules as of December 2020 as those were approved as well. Scans and results. You can see on this graph here, the midline shift. This is after 2 years on an MRI here of treatment, and the midline is back to normal. And there's only a small area now where there's a small cavity where the tumor used to be. What is the personal impact on me? This drug has saved my life. I can unequivocally tell you that this impact is unlike anything that was out there. My alternatives were not great, and it was just a process of waiting for the disease to return. A comparison. You can see here the angry tumor on the left, again about the size of a large egg. And then now, this is the same area over here after 2 years of treatment. So very effective, lots of things. 1% in the future. Yes, it's only 1% chance, but 1% is so much better than 0. And I can honestly tell you right now, if there's 100 people watching, just statistically, one of you is likely to be the person that would have this if you've got a solid form of cancer. And out of there, all the doctors and oncologists that are watching right now, how many doctors are watching right now with how many patients with any form of solid cancer? Get people tested. This is the future of medicine. No matter how up to date your oncologists and doctors are, they simply can't know every new drug or treatment. Research and become your own best advocate and work with a receptive oncologist. Personal impact. What can I do? As I've stressed, be your own best advocate. I want to help others. Some of the things that I've done the Keto for Cancer diet with calorie restriction and intermittent fasting. I've been on that for 2.5 years. I took Boswellia while I was undergoing my brain radiation, is because it always increases brain swelling, and almost everybody has to go on dexamethasone. I didn't have to because I was taking Boswellia. I did hyperthermia treatment. I do exercise. I try and stay at peace, have some relaxed time, do walks, spend time with family and friends, and have some quiet time as well. All this is important. I've also created my website, Glioblastoma Guide, where I have books, videos, resources, and blogs that I've written that condense all the material that I've learned, because patients with glioblastoma have a very short life expectancy. So instead of everybody having to do all this research, I've tried to put it all in one place so that they can activate it and talk it over with their doctors, their medical team, their family and friends, and make their best decisions going forward. I also have a YouTube channel, Glioblastoma Guide, where I'm doing the same kinds of things, but in a video format. I'd like to thank Bayer Healthcare Pharmaceuticals, my oncologist, my pharmacist, surgeon, support of my family, friends, relatives, and loved ones, all those who have helped make Vitrakvi possible, including you, Dr. Poul Sorensen, which is part of my miracle in the making. Thank you.
Great, thank you all so much for your respective presentations. Those are all very informative. So we still have about 10 minutes, so we're right on time. And we have some time for audience questions. If you haven't already submitted your question, you can still do so, and hopefully we'll be able to get to it. So our first question is, would this treatment be used for uveal melanoma with monosomy 3 if it spreads?
I think that as we saw in basket trial, and the whole philosophy of tumor agnostic treatment is such that it doesn't depend whether it's a uveal melanoma or pancreatic cancer. As long as the gene fusion is positive, we have a target, and then Larotrectinib can be used.
Yeah, get tested.
Great. Our next question is, if a patient wanted to get genomic testing done, how would they go about it? Do they have to meet certain criteria before they can request that?
Dr. Sorensen, do you want to answer it, or should I go ahead? Oh, okay, all right. So I think that for what we are doing here in BC is a process which may not be available in other jurisdictions, but talk to your healthcare provider and they may be able to guide you how to go about getting tested. We have a fast track program by Bayer where we just have a permission from the patient and get this sent over to an accredited lab for get the NTRK testing done. And the other method is that Ted has taken, which will be Foundation 1. Ted, can you go ahead and elaborate that process for us as well?
Yes. So in that process, what happens is you actually do an application up with Foundation 1. When you do that, they will send in a group of paperwork. Your doctor fills some of it out, your oncologist, and then the next step is they actually get a piece of your tumor, and that is flown specially to their lab, and they undergo the genetic testing there. Now, to speed up the process, just as a tip to anybody who might be doing this, we actually took the form from the oncologist, and I knew where they were keeping my particular tumor was at the location where I had my surgery. We actually went right to the facility, right to the area, and gave them the paperwork directly. They were able to go downstairs and do it that particular day. So in order of magnitudes, time-wise, because sometimes bureaucratically in hospitals and whatnot, things can get bogged down a bit and you get put on the bottom of the pile. That was just something in my case with glioblastoma that I wanted to make sure that we were doing this as quick as possible. So that would be the process there. And it only took, I think, about 2.5 weeks or so, and we received back the report. It's about a 38-page report, very detailed, and it will list on there not only clinical trials that may be applicable to your case, but also any of the particular results of your genetics on your tumor, including any of the mutations like EGFR and things like that. And it will suggest the medications. In my case, it was Larotrectinib and possibly Entrectinib if needed late down the road.
So sorry, I was muted before. Apologize for that. So in answer to this question, one of the difficulties has been setting up a screening algorithm across Canada. And some of us pathologists have gotten together to write some kind of white papers on how we think this should be dealt with. Because one of the things that terrifies the government is that they'll have to pay for screening for NTRK fusions across many different tumor types, and they're terrified of the costs for that process. So if, for example, it's a congenital fibrosarcoma or a secretory breast cancer where the incidence is super high, towards 100%, then it makes complete sense to do the testing. But the worry is in something like glioblastoma multiforme or colorectal carcinoma, in which the incidence might be 1%, it's very difficult to figure out a way to decide when to test for it and when not to. So the process is still kind of in play, and foundation medicine is a very good way to get around this. But of course, the patient has to pay for this themselves, and so that's wrong. So we need to figure this out. We need to figure out a way that we can decide on a system for diagnostic intervention across the whole country that is paid for by our healthcare system, as opposed to putting the onus on the patients to do this. And we're working on this. But that's been one of the issues with adoption of. It's one thing for a drug to be approved. It's another thing for the government to actually pay for it. So be aware that even if a drug is approved, it doesn't mean that the government will pay for delivery to the patients, even though, as Ted beautifully illustrated, it can have profound implications for patients. So this is something we still need to work through. And I think advocacy work like you're doing, Ted, is the way to really push this across the finish line.
Thank you. Yeah, and I can explain that it did cost about $8,200 to get that testing done. One thing, just in some of my research that I can say is that I know the sophistication of DNA analysis is getting better and better and quicker, which should drive those costs down over time as well. So that's just another comment.
One of the ways that we can diagnose NTRK fusions, or where we're trying to do it, is to use something called immunohistochemistry, where we use an antibody against what we're looking for. And this is something all pathology labs can do. The problem is if it's not as sensitive or specific as it needs to be, and so that's still being worked out. But that would solve all of this, because then a biopsy could be submitted to a routine pathology laboratory in any hospital in British Columbia, or practically any hospital that has a pathology department, and then they could determine whether there's an NTRK fusion without needing to sequence the sample. And sequencing is obviously more difficult, and not every center can do it. Then it would probably need to be centralized in the Lower Mainland, for example, at the Genome Science Center.
Great. Our next question is, what impact do you think that the approval of Vitrakvi will have on the approval of other future tumor agnostic treatments?
I think that's a really great question. And, just to be brief here, I think that's why it's been so hard to get this approved, because it's a very different way to think about things. When you come in with Herceptin to treat breast cancer, that's something that the regulatory bodies can understand. But now you suddenly have a drug that is tumor type agnostic, and I guess that needed to get approved, and I think it'll pave the way for other agnostic drugs. That's my impression.
And our next question, oh, sorry, is someone going to jump in? Nope.
Nope.
Our next question is for Ted. So aside from the issues with getting your genomic testing done, were there any other significant barriers that you encountered on your path to treatment?
There definitely were. There's whole parts of this that are often sort of overlooked almost, but there's a whole side of this where your whole world is turned upside down. When you get a diagnosis like this, it's not like something else, like where you break your arm or something. It's like there's a clear path to what's going to happen next. It's all brand new, usually to the patient. They're not familiar with this chemotherapy. They're not familiar with radiation therapy, the types of decisions that you're going to have to make on a quick basis. And then on top of that, some of your research and reactive results, because the standard of care is just that. It is the standard of care. And a lot of doctors are kind of locked into it, and it is a wonderful thing, but it does have its limitations. And unless you're kind of pushing forward on things, we're not going to make advancements. So, some of the things that I've done, for instance, that Keto for Cancer diet, that is a restricting both glucose and glutamine and restricting calories. And the research that I have on this, you can see some of it in my website and in Miriam Kalamian's book, Keto for Cancer. These are just things that people can do regardless of the type of cancer. But I've found that this paves the way for some treatments that are not necessarily front and center, but are going to possibly make a real difference in what you can do. But I can't stress enough to be your own best advocate in this process, because without that, you're really just sort of in the current on an inner tube floating through the lazy river ride, and take a paddle and do something. That's what I would recommend.
Great, thank you, Ted. And we'll do one more question. This one is directed for Dr. Zulfiqar. So when treating patients with rare forms of cancer, how do you go about ensuring that they have the best treatment methods available?
I think the usual standards are that you look at the highest level of evidence out there to be able to treat them. And then whenever there are some cutting edge or any novel treatments available, our first attempt is that to utilize and get benefited from them and under the umbrella of clinical trials. Because whenever we have novel treatments, according to human ethics, it's not appropriate to just offer it based on the preclinical hypothesis or information, even though if that information is published in a very prestigious journals. One example, Dr. Sorensen, you might remember, is that the DCA being, I think it was a few years ago, University of Calgary had this basic science published in journal Nature, where dichloroacetate sort of changed the mitochondrial metabolism and promoted apoptosis, and it looked very promising. in basic science aspect of it, but it didn't translate the same way into the clinical trials. And then therefore, the short answer to that question is, number one, find the highest level of evidence, which are usually phase 3 clinical trials, and look through clinical, currently available clinical trials, and then people like Ted who are highly motivated and their own best advocates are the ideal type of person to work with in a healthcare team as you become a healthcare partner rather than an advisor only. So I think that it's a teamwork where we can work together to get the best possible outcome.
I agree, and I would just add to that we've established something at UBC to really look at this question. How do we, what do we have to do as a team, including the whole pipeline of involvement from researchers to clinicians to patients? How do we work as a team to shorten the time it takes for some of these discoveries to make it over the finish line, and to improve the chance that a discovery is properly kind of vetted along the way to actually prove benefit. As you just said, Dr. Zulfiqar, there are lots of examples of high impact publications really not standing up once you get out of the mouse studies into human studies. So we really need to develop a pipeline where we can facilitate the whole pipeline of translational delivery of these discoveries, make it faster and get it to the patients faster.
Mm-hmm.
Well, we're just after 2 o'clock, so I think we'll begin to wrap things up here. So thank you all so much for taking part in this presentation today. You were all incredibly informative, and I know our audience certainly appreciated each of your unique perspectives on this very important topic. So thank you everybody for attending and joining us for today's webinar. Take care, stay safe, and we'll see you soon.
Thank you.
Thank you, Layla.
Bye.